Imagine a world where the damage from a really devastating neurological disease, well, where it couldn't just be slowed down, but actually reversed. For so long, the focus with things like Parkinson's has mostly been managing symptoms, maybe delaying how fast things get worse. But what if we're like right on the edge of something much bigger, something that hints genuine repair might actually be possible? Welcome to the deep dive. Today, we are plunging into a truly fascinating new study on Parkinson's disease that offers some unprecedented hope. Expert, this research is definitely creating some buzz. What's the absolute core of this breakthrough, and where's it coming from?
Well, what's really interesting here is exactly that change you mentioned, that shift. Moving beyond just, slowing down the inevitable progression to actually thinking about restoration. And this remarkable work, it's titled Reversing Parkinson's, Enzyme Inhibition Restores Brain Function, just came out in Science Signaling, July 1st, 2025. It's from a fantastic collaboration between Stanford Medicine and the University of Dundee. And it really does give us this tantalizing glimpse of how restoration might actually work.
Restoration. That is a powerful idea. So for you listening, our mission today is, to unpack the specifics of this really groundbreaking research. We want to understand the science behind it, and it gets pretty intricate, and explore what these, quote, astounding results could actually mean for the future of Parkinson's treatment. Okay, so let's unpack this. What exactly is at the heart of the problem they decided to tackle here?
Right. At its core, this study zeroes in on a specific genetic mutation. It's actually a pretty common cause of Parkinson's, maybe responsible for about 25% of cases. And this mutation leads to an overactive enzyme called LRRK2. That's LRRK2, leucine-rich repeat kinase 2.
Yeah.
You can kind of think of it like a vital component inside cells that's just gone a bit haywire. It's stuck in the on position in certain brain cells when it shouldn't be.
Okay, LRRK2. And when it goes into overdrive like that, what exactly is it doing? How does it cause damage in the brain? What's the mechanism?
So when LRK2 is excessively active, it basically disrupts the structure and the communication lines within brain cells, fundamentally. It has a particular impact on the dopamine-producing neurons. These are obviously crucial cells, especially in how they connect with a part of the brain called the striatum. And the striatum, it's this deep brain region that's heavily involved in how we move our motivation, even decision-making. So that's hitting a critical hub.
So it's sort of like a major communication breakdown right in a vital control center of the brain, affecting all those things we rely on daily.
Exactly right. And you can see how the implications for just, daily function are pretty profound when that system starts to falter.
Okay, now here's where it gets, I think, really interesting. Looking closer at the cells, how does this LRRK2 disruption actually, show up at the cellular level? What does this breakdown look like up close?
Well, this overactive LRRK2 enzyme essentially makes brain cells lose something called their primary cilia. Think of these primary cilia as like tiny but vital cellular antennae.
Antenna?
Yeah, antenna. Cells use them constantly, sending and receiving critical chemical messages, so losing them. It's effectively a communication blackout. The cells suddenly become isolated. They can't talk to each other properly.
Okay, so if these critical antennas are down, what are the most important signals, the key messages that are suddenly just going unheard?
One incredibly important pathway they're tied into is called the sonic hedgehog signaling pathway.
Wait, Sonic Hedgehog, like the video game?
Yes, exactly. Named after the cartoon character, surprisingly. So in a healthy brain, when dopamine neurons feel stressed, they release this sonic hedgehog signal, specifically in the striatum. And this signal, it tells certain neurons in the striatum and also these support cells called astrocytes to start producing neuroprotective factors. These factors are crucial. Think of them as the brain's own little first aid kit, designed to shield cells from damage, stop them from dying prematurely. But here's the rub. When those striatal cells lose their primary cilia, because LRRK2 is too active, they just can't receive that sonic hedgehog signal, which means those vital neuroprotective factors, they just don't get made. And it's actually a devastating kind of double hit, because not only are they missing out on those protective signals, but the very cells that lose their cilia, they are themselves put on a path towards dying. They actually need those cilia to receive other signals that keep them alive and healthy.
Wow, okay, so it's not just that they can't get help from the outside, but their own lifeline, their own communication for survival gets cut too. That really is a cascade, isn't it? A failure at such a basic level.
Precisely. And that really sets the stage for the kind of progressive degeneration you see in Parkinson's.
Okay, so understanding that problem so deeply, what was the scientific approach to actually fixing it? How did they try and restore this lost communication, stop this damaging cascade?
Well, the researchers introduced a specific molecule. It's called MLI2 LRRK2 kinase inhibitor.
Uh,
This molecule, MLI2, it's designed very specifically to attach itself to the LRRK2 enzyme. And crucially, its job is to decrease its activity, bring it back down towards normal, healthy levels.
That sounds incredibly targeted. Did it work right away, or was there a bit of, trial and error before they saw these amazing effects?
That's a really good question, and it does highlight how persistent you often need to be in research. Initially, they actually gave this inhibitor to mice for just 2 weeks, and frankly, they didn't see any significant changes. It must have been pretty disappointing.
Yeah, I bet.
But then they drew inspiration from some other recent findings. These were about non-dividing neurons, different ones involved in circadian rhythms, our sleep-wake cycles. And the primary cilia on those particular cells, they were actually observed to grow and shrink regularly, even though the cells themselves weren't dividing.
Oh, interesting.
Right. It suggested that maybe cilia could actually regrow on mature neurons if you gave them enough time, that they weren't just permanently lost once gone.
So they thought, okay, maybe we just didn't treat them long enough. What happened when they extended the treatment period then? What were the results?
The results were, well, in the words of Dr. Suzanne Pfeffer, one of the lead researchers, they were astounding. After 3 months of the mice getting this inhibitor in their food, the percentage of striatal neurons and glia, those support cells that had primary cilia, it was indistinguishable from healthy, normal mice.
wow.
They'd effectively regrown their antenna. It's like going from a really patchy mobile signal area back to having full bars. The communication lines were open again. And that finding itself, it really challenges a long-held idea about mature neurons, suggesting maybe other kinds of damage we thought were permanent. Maybe they're also fixable if we find the right pathway.
That is genuinely groundbreaking. So, okay, beyond just the cilia growing back, what else did they see in these treated mice? What other positive effects did this restoration trigger? And what does it all mean for the bigger picture?
All right, this is where it gets really exciting because the cilia regrowth wasn't the end of the story. It was just the start of this remarkable positive cascade. First, those striatal neurons and glia, they were once again pumping out those vital neuroprotective factors, responding properly to the hedgehog signaling. Also, the hedgehog signaling coming from the dopamine neurons actually decreased, which sounds bad, but it's good. It means those dopamine neurons were under less stress. They didn't need to shout for help as much.
Okay, that makes sense.
And maybe the most significant finding, indicators they measured for dopamine nerve ending density, like how many connections there were within the striatum, they doubled. Doubled. Which means those crucial communication lines from the dopamine neurons, the wires, if you like, weren't just being protected. They were actively regrowing, making more connections. It suggests real, tangible restoration was happening. And the implication there is profound. It suggests it might actually be possible to improve a patient's condition, not just stabilize it or slow the decline. This really moves us beyond just managing symptoms. It points towards potentially being able to, rewind some of that early damage.
That is a truly massive shift in how we might think about this. But, the big question always is mice to humans. That's a huge leap, right? What are the biggest hurdles or maybe the potential pitfalls that researchers are really aware of in trying to translate these incredible findings into actual patient care?
That's absolutely the right question, and it's always the critical step. The primary hurdle, well, like with any finding from animal models, is making sure these effects translate safely and effectively to people. Human physiology can be quite different. Researchers have to be really careful about potential side effects, what they call off-target effects of the inhibitor, making sure it doesn't mess up other important processes in the body.
All right.
And then there's getting the dose right, figuring out the best way to deliver it in humans. There's still a very rigorous clinical trial process ahead to prove both efficacy, that it works, and crucially, that it's safe.
That makes total sense. So keeping that in mind, what are the sort of practical implications for patients down the line? How might this change how we approach Parkinson's, especially thinking about, say, early intervention?
Well, one of the biggest practical implications could be in early detection. It's interesting. Parkinson's symptoms can actually start maybe 15 years before the tremors that most people associate with the disease appear.
15 years. Wow. Like what?
Yeah, things like losing your sense of smell, chronic constipation, sometimes a sleep disorder where people physically act out their dreams. These can be really early signs. So the hope is, if people who carry that specific LRRK2 genetic mutation could be identified much earlier, maybe through genetic screening perhaps, they could potentially start treatment with an inhibitor like MLI2 as early as possible, before too much damage has already happened. That would shift everything from being reactive to being truly proactive.
That really would be a game changer, catching it that early. So what are the immediate next steps for this specific research team? Where do they go from here?
Okay, so their immediate next step is to really rigorously test if other forms of Parkinson's, the ones not directly linked to that LRRK2 gene mutation, could also benefit from this kind of treatment. That's crucial because even in other forms, LRRK2 activity might still be abnormally high, so maybe the inhibitor could help more broadly. And it's also important to know there are actually multiple clinical trials of different LRRK2 inhibitors already underway for Parkinson's. So the hope is that these really striking findings in mice will help inform those trials, maybe accelerate things, and hopefully translate successfully into benefits for human patients. And, the study itself had significant backing from major organizations, the Michael J. Fox Foundation, the Aligning Science Across Parkinson's Initiative, the UK Medical Research Council, which really shows how important this line of research is considered.
This deep dive has really given us, I think, a genuine aha moment. What we've seen is that by targeting just one specific overactive enzyme, researchers managed to restore vital cell communication, actually regrow those tiny cellular antennae we talked about, and essentially reignite a protective system in the brain. And that really does move us beyond just slowing things down towards the potential, at least in mice so far, of reversing some of that early Parkinson's damage. Understanding those mechanisms, those antennae, it somehow makes the potential for recovery feel much clearer, much more tangible. It offers maybe a new kind of blueprint for tackling these kinds of diseases.
Absolutely. And maybe here's a thought to leave you with. Consider this. If a single targeted molecular intervention can revive fundamental cellular processes like this, even fixing structures in the brain we thought might be permanently broken, like those cilia in mature neurons, well, what other lost signals might we be able to discover and potentially restore across all sorts of other conditions? Maybe just by getting better at understanding the subtle, incredibly intricate dance of our own biology, what else might be fixable that we currently think isn't?